All right.
Can you guys see my screen?
Oh, not yet. Can you unshare, Leonidas?
Can you see it?
Yeah, we can see it. We'll put it on in a minute. We'll do the intro first.
Okay, yes,
Yeah.
sure.
All right, thanks. Yeah, can you unshare?
Can you unshare? Yes.
Yeah, can you stop sharing? There we go.
I didn't get that.
We'll start you. All right. Okay, well, hello everyone. So welcome to our final Dean's Digital Cafe of fall 2003. I'm William Letzu, an assistant professor in biological and chemical sciences here at the College of Arts and Sciences here at New York Tech. On behalf of Dean Quigley of the College of Arts and Sciences, I'd like to thank all New York Tech staff, students, faculty, staff, alumni, and guests for tuning into our event today. So I'd like to remind everyone that today's talk is being recorded. We'll be taking questions following the presentation, but you should feel free to enter questions or comments into the chat at any time. I'd like to also invite you to join our Dean's Digital Cafe Canvas course, the link which can be found here. Oops, not that link. The link which I'm going to post in the chat right now. Where you can go to watch recordings of today's and past events. So before introducing Dr. Salichos, I'd like to acknowledge the land. The Long Island campus of New York Tech sits on a larger territory known as Pominoke, which was home to numerous indigenous communities, including the tribes of the Mattincocks and the Merricks. I'd like to acknowledge the land, the stewardship, and sovereignty of the native peoples and their enduring importance, even in this virtual environment, to our work and community here today. And now I'm pleased to introduce Dr. Leonidas Salichos. So Leonidas joined NYIT as an assistant professor in biological and chemical sciences in 2021 and is currently based at the Manhattan campus where he teaches biostatistics and bioinformatics. So Leonidas did his PhD work at Vanderbilt University where he studied phylogenetic trees using machine learning techniques, and as well as completed a postdoctoral fellowship at Yale University under Mark Gerstein. He's published numerous articles on driver mutations in tumor evolution, on the spread of COVID-19, and even the evolution of music and culture, some links to which can be found here. Leonidas leads a vigorous group of undergraduates who are continuing to produce research in the fields of phylogenetics, next-generation sequencing, and cancer immunology. In addition, and which you'll hear about more today, Leonidas has been recently named the director of the new NYIT Center for Biomedical Data, which keep an eye out for the announcement. So welcome, Leonidas. Thank you for joining us at today's Dean's Digital Cafe, and we are excited to hear more.
All right, thank you, William, for the great introduction, and hello everyone for the talk. I would like to welcome you at the cafe as well. Let me try to share here. So can you see the screen?
Looks good.
All right. So all right. So I'm going to be talking about today about the using pretty much evolving systems that I've been working on from personalized medicine in cancer and precision to how different models that we try to have in the pandemic in order to be better prepared or like have better mitigation strategies. I'm not going to talk about my whole work. I'm just going to focus on this kind of like two type things. I'm going to start with a little bit on cancer evolution, and then we're going to discuss on viral evolution. We're going to have some questions at the end. Feel free to reach out if you have any more questions after the talk as well. Let me start with this. And yeah. All right. So most of this work that I'm going to be presenting today happened around like 2018 to 2020. I finished it here and at Yale. So it's a little bit in cancer. So we had a lot of we're part of the PCOG pan-cancer analysis of whole genomes. That was a huge consortium with many researchers. And we had about 2,800 whole genome tumors that were sequenced. and they were really well sequenced. So yeah, so just a small introduction what I about cancer and evolution of cancer. So we normally talk about cancers when we refer to drivers in cancer. We normally mean like the 1 or 2 or 3 mutations that did a big thing, and then after this, the cancer started evolving. But in reality, normally, each cancer, if you sequence it, it has hundreds to thousands of mutations. And this can be a long range from like really important mutations to actually even mutations that don't matter that much in cancer, or they can even be negative. So this is kind of like a level of selection that happens within the cancer environment that can range from all the way to like strong negative selection to strong positive selection. Obviously, this is for the benefit of the cancer, right? So there's not going to be a lot of deleterious mutations because they're going to be washed out. There are going to be a few really strong drivers, and then there is a range of mutations in between that might help or facilitate the cancer. So just to explain a little bit, you probably know some of this already. What happens is you have a normal cell at the beginning, then that cell gets hit by a lot of mutations. Some of them might matter, and once, let's say, you get one driver mutation, then you start having what is called like a subclonal evolution. So based on the different mutations, you can have different parts in the cancer that kind of like grow, and then each of them might have different mutations that are important for the cancer. Now, when we sequence a cancer, a tumor, pretty much what we get is a list of all the mutations you can find there. And then we also have what is called the VAF, which is the VAF allele frequency. So this is kind of like the percentage of like in the population, how often do you see that mutation? And then it's obviously, if a mutation happened early, it means that more cells would have that, while if a mutation happens later, it means that few of the cells would have that mutation as a general rule. So based on this, I first started to observe different categories of mutation, whether they seem to be enriched in happening early versus late. And then obviously the driver seems to happen. You can have drivers later, but for the most part, it's like if you have a known driver, you can obviously observe it to be an early mutation. And then I was looking at different other categories. If it's a high impact coding mutation in the organism, it also seems to be earlier. And then there's somewhere in between that are happening. What is interesting, you can see, for example, here the tumor suppressor mutations are really happen early because this is a big advantage for the cancer. The cancer is not suppressed anymore. And then the same thing with apoptosis, it seemed to be happen early. What was a little more interesting and kind of like counterintuitive, if you think about it at first, it's like for oncogenes, it wasn't like positive or negative. It was kind of like about the same. And this is because the mutation in oncogenes are a little more complex, because in oncogenes, you do not want to knock out the oncogene. These are like big hub genes. You just want to switch its expression. And the same thing with DNA repairs was like often people think that, oh, you have a DNA repair mutation, it's important. Sometimes it is, but for the most part, if you have a DNA repair mutation, kind of like knocks out the cells. Like you cannot have a cell that just like create without any repair mechanism, just like evolve. So this is kind of like the results that we had from this big consortium with 2,800 tumors. And from there, I got like my parallel work was also to see if we can, based on the frequency of these mutations, to see if we can detect the impact of these mutations and also how the tumor was growing. So pretty much you're looking at that list at the end, and then you try to make predictions how the tumor evolved in the organism. So I developed a framework that it's like with efforts to be able to detect drivers during like a tumor progression, calculate the effect, identify the growth of the pattern, and then provide insights into the tumor evolution. The difference in this is normally to detect the impact of mutations, you need thousands and thousands of tumors or hundreds. But for this case, my interest was to be able to model this in the way that you can detect these just by looking at a single tumor. So just by a single sample to be able to detect where what the mutations are. This is kind of like I call the stories 1001 tumors, because at first I was dealing with a large sample of like about 1,000 tumors, and then I focused the research on just one that was like really deeply sequenced. That means that we had a lot of very much accuracy in the frequency of the mutations. Just a few words. I'm not going to tire you with too many details or obviously the math of this. So what happens is at first you can see the different frequency of the mutation. If you have a tumor that is just growing regularly, just a simple exponential growth, or like a population, and then you have what happens if at some point you hit a specific mutation that it's now it has a higher fitness, so these cells tend to replicate more. And this has an impact of not only that mutation itself, the one that is denoted as like this calligraphic F, but also the mutations that happened before this one. So for example, the red and the blue one, you see that also increasing. And the reason is because this population, as this population increases, it also increases the frequency of the mutations that happened before that and exist in the cell, right? So based on this observation, I calculated what is the expectation of change, and I use this what we called generational hitchhikers. So these are mutations that are hitchhiking, and then they're also getting really prevalent. And also, since they happened earlier, they're going to have a higher mutation than the one that is actually has the frequency. Again, I'm not going to go into the math. This is kind of like the equation that I modeled about. And then in reality, what I'm pretty much trying to model is the difference. So if you consider the different mutations in line in order from earlier to later, I can calculate in big chunks like the expected difference in the frequency and see and pretty much model the effect of increase or decrease based on the different frequencies. To demonstrate this, let me show you, this is on a simulation, how it works. We have simulated a tumor growth with a mutation that has an effect of 3. Here we order the mutations from earlier to later. And if we apply this model, you can see on the top graph, the growth of the population, and on the bottom graph, you can see the impact. So you can see, obviously, there is noise that you always have to deal with and model it. But at the same time, in that scenario, we can pretty well and accurately detect this is like the impact of the mutation and also how the tumor was growing throughout that time. And this is a simulation. So this is kind of like where the position of our predicted driver. Obviously, we can calculate a window and pretty much see what mutations fall within that range and be able to make suggestions about these mutations. This is based on our simulation. Obviously, the better sequence the tumor, the more accurate we are by being able to detect the effect. But we get a differentiation between the different mutations and their effect early on from around 300x. For those that don't know what it is, it's like when you sequence, it's like how many times do you expect to find a specific position in the genome sequenced in a way. So that 300x means that you expect to find it 300 times when you're mapping the genome. So first, I took that to like this 1,000 tumors. This is one example of them. Obviously, the quality of the frequencies here is fairly low. It's normally close to 70 to 100x. So there's a lot of noise in these tumors. And this example here, we know from Peacock, we knew the drivers of that tumor. And you can see that it kind of like corresponds to the growth patterns that we're calculating. So we have an initial growth, slowed down, another growth, kind of like negative here, but then we have a third one that kind of like, again, hits the growth of the tumor. This is kind of like without averaging with all the noise, you can see a lot of like negative and the positive on the bottom. But at the same time, we had a lot of actually a very large percentage of the tumors that we're dealing with. We don't really know what the driver is. So in this case, you can actually see, so that was a case where Peacock was not able to find a driver, but then you can see where you can look at around. So you have some sort of like positive growth at the beginning and again, like a pickup later. So you can estimate, you can pretty much let's assume that there are at least two drivers in this tumor. So yes, so these are like suggestive ways. Overall, there were like three different patterns, as you can expect. So there is like the red ones are the ones that are like kept going. They're pretty aggressive ones. The dark one, they kind of like plateau at some point. They kind of like really grow at the beginning and they kind of like plateau. And then the blue ones are the ones that seems to be either artificially from the data or not be growing as much, kind of like start decreasing at the end of their not the end, like when they're sequenced. And obviously this doesn't consider metastatic tumors as well, which is like a different animal in this case. So by looking at specific categories, just to get some confirmation on the experimental data, you can see here when I tested the peacock drivers, they all seem to be so like not even like even with the noise, they all seem to be in positive growth, estimated and reached in like periods of positive growth. The Vogelstein genes, which is like cancer genes, is a set of both and tumor suppressor genes. There was like kind of like enriched. This is significant. And then the random genes were not really. There was kind of like all over the place. When I looked at like the different mechanisms that seem to be mostly hit by mutations that belong to like positive growth, I think that stands out, and I think it's a nice narrative. Is like that the first thing that comes out is like the multicellular organism development, and I think that makes sense if you think of tumor, that the first thing that the cell can get rid of is like they don't really care about acting as an organ anymore or any tissue communication. It becomes more like a more selfish game. So it's easy for them to get rid of all this kind of like multicellular development or like communication between different cells. Obviously, you can see cell adhesion, cell development, cell differentiation. These are like different mechanisms that are getting hit. And then we can go to when we go to more specific mutation, we have a different mindset for tumor suppressor genes and oncogenes. As I mentioned before, if you're a tumor suppressor gene, for tumor to do well, you need to be knocked out. If you're an oncogene and you're being knocked out, then the tumor is not going to do well because it doesn't grow anymore. So, and you can see that from the significant enrichment between these two. If you're a tumor suppressor gene, you're more likely to be in a positive growth if you're like a promoter in the promoter. If you're a nonsense mutation, a missense, or an intronic, it does not care that much about synonymous mutations. On the other hand, if you're non-oncogenes, like the two things that really stood out, it was the promoter region, and the synonymous mutation was surprising because until like people don't really think of synonymous mutation to matter in tumors, even though obviously the effect doesn't have to be too big to be significant here, and you can observe this. So for example, the tumor suppressor genes, you can find intronic mutations that might matter, and that can be justified if you can actually have a splicing effect that can knock out a gene, a tumor suppressor gene. And this obviously these results really pair really well with what I was explaining before, where you can see the nonsense mutation in tumor suppressor genes that they relate really well. In terms of oncogenes, you can see the missense mutations that they don't really seem to be enriched in early mutations. It's kind of like a more mixed effect. And then from some Pappel's data, you can see also that how this corresponds to missense mutations in terms of tumor suppressor genes and intronic. Anyway. So let me focus a little bit more on what I was talking about, synonymous mutation. So these are like obviously it comes down to how much data you're dealing with, but these are different type of mutations that we're able to like see how they are enriched or not. And you can see like the TP53 promoter. These are like known cancer genes as well. What really stood out is also the BACL2 synonymous mutation that is really high among these genes. And if you look at the profile, you'll see that most of the mutation kind of like are focusing in this specific region where you can have like the promoters and the introns. And what's interesting about this is was like the exact same type of gene, which is the BCL2L12, which is like an ortholog of that gene. There are studies that suggest that synonymous mutation matter, and the reason is because they kind of like change a specific sequence where you have a microRNA binding to that kind of like affects the regulation of the gene. And remember, for oncogenes, it's more like a fine tuning rather than knocking in like increasing or decreasing that specific gene. All right, so let's focus a little bit now on this on a single tumor, as I was saying before, which is an ultra deep sequenced amyloid tumor. The other thing about amyloid tumor is that you have less things to worry when you're like sampling because it's a liquid tumor, so everything is there. It's more, the frequencies are more representative of like space. So based on what we're looking at this study that was done, we already had 3 driver mutations, but they kind of like really fall into this positive growth for the tumor. Although there's also a positive growth at the beginning that doesn't seem to correspond to this. And there, based on the data set that we analyze, there seems to be some other like mutation that could actually correspond to this specific initial growth, and then the tumor kind of like slows down, and then you have like this final hit that after this is like you have like the growth of the tumor for a long time. In green, we have like the known drivers, and then in yellow you can have driver candidates. And it's really, really impressive if you can see from the algorithm how these drivers really corresponds to this specific like peak, long peaks of like for the growth and the effect, which is, to me, it was really surprising when I saw it, like this kind of like precision. All right, so the few things about the future direction in this is like, obviously in personalized medicines, like you can actually look at specific tumors, but they have to be well sequenced when we're doing this. You can apply this to cancer liquid biopsies. You can study negative selection, so perhaps take advantage of specific mutations. I'm interested in developing a better quality software for people to be able to use this, because now it's still at the level of like scripting, and then expand it to a couple of other sequencing data sets. And I think that's also like useful to also understand the tumor evolution in terms of like fitness for the different mutations. All right, so let me take a breath here and like switch the subject a little bit. So that was about the precision medicine in tumor and how we can actually, based on the specific mutation, the personalized individual, to make assessments about the way the tumor has been growing, how aggressive it is, and build a profile for the tumor. I want to talk here about another thing that I'm working on, part of my research, but has to do with viruses. I started working on this when COVID first hit, like a lot of people. I do have a background in like studying evolution and viruses, so I wasn't like just starting out of nothing. But I had an idea that it was how to be a little more precise in the way that we try to develop strategies to deal with the pandemic. And I will explain how that came about. So this is pretty much what we have as a data set when we're talking with viruses or pretty much any organism. This is this is the DNA. You have here all the different strains on the left, and you can align them and see the positions. In this specific case, these gaps means that there either is a deletion or we don't have enough information. So this is kind of like the data that we have, right? And then from this data, we try to pretty much predict and like extrapolate all the other information of analyzing. Now, how we do that? So we can take, let's say, for example, a random tree, and then the question is how well the data fit to that tree, right? So in a way, we pretty much will have to explore a huge number of trees with different algorithms and find the one that fits this data first. But at the same time, we need to make some assumptions. And you can also like need to estimate the distance between the different branches and the different species. But before moving from the data to the tree, we need to have an evolutionary model. And that has to do, for example, with the different frequencies of the nucleotides or the amino acids, the probabilities from changing, for example, from A to C or from C to G. These are different changes that can happen and don't necessarily have the same probability. Mutation rates, for example, different organisms can have different mutation rates, and also consider mutation rates across the whole genome and different invariant or variant size.
Yeah,
So all of these are like parameters that we have to integrate into our evolutionary model to go from a data set to a tree. At the same time, when we're dealing with viruses, there are also other things that we really need to consider that, for example, if the population is increasing or decreasing, can really matter for like the shape of the tree, right? If this is the left tree, here is like for a constant population. This is the right tree is like you see for the population that it's expanding. That is the branches are like shorter. And other stuff are, for example, you can have a bottleneck that can also really, this is the same data set that we have, but even considering a bottleneck can really change the shape of this tree. So one of the other things that matters, like also how the population is changing to be able to calibrate. So what happens is we have the data set, we have our model, all the different the models that we can for mutation rates, and then we go to that whole tree space and we try to find the best tree out of there that fits our data. Obviously, there are like some optimization algorithms. It's impossible to look at the total tree space, but there are ways of like navigating through and like find a really good tree. And then if with Bayesian inference that we're using in this case, we can actually also include like geographical model, population growth model, etc. as well. So pretty much it's like looking at the space to try to find where, like in different peaks, try to find a good tree. This is a general introduction to phylogenetics. Hopefully it's going to be a little more helpful because we're going to be looking at some trees. So the whole idea was, if you remember the pandemic, initially we had two main hotspots. And then people seem to treat the same thing, all the virus throughout the whole US. My way of thinking of this was more like a wave or an outbreak that keeps moving around a geographical map. So this means that the distance between one spot and the hotspot that initially was created was really important, especially at the beginning. So I started looking, so you can see here like that it matters where you are from the initial where the virus is initially, how it spreads, and then you can estimate that distance and again sample across different time points to see how the virus is spreading anyway. Another measure that you could use, I don't know if you're familiar with this, or it's kind of like it's a trigger alert for like a lot of us, was like we're just observing the death rates and the infection rates throughout the pandemic. You can actually always measure, like sample the number of deaths per 1 million population as a way of like knowing how bad the virus is doing and how it's spreading across time, right? So the first thing that you could really see immediately was how these numbers of deaths were relating to like where you were based on the distance from New York City. And this is also if you include as a second hotspot the San Francisco, or it can be in Washington. The only point that was really off was like New Orleans, that are kind of like doing their own thing back in the time. But other than that, you get a pretty good fit for like a curve based on the distance from the pandemic. And people are not really talking about this back then. It was like everything was treated the same. The other implication of this is like you could be like South Carolina and North Carolina, and then you can get hit twice from the wave, either at the same time or from different time points, right? So like it's not necessarily a linear thing. So my approach in this is to build kind of like a network to see if we can predict the viral spread and also see whether we can actually intervene in that network. I'm going to present a little bit a paper that was recently published, that I finished it here, but I was also started when I was at Yale. So initially we kind of like sampled sequences from all over the world. This is like with the ones that we used as reference sequences, and then it's kind of like span the whole the COVID pandemic. What was good at the beginning was at some point, well, it's good. It was not good, but the first few months, it's a good model because, for example, a lot of the flights were not happening. They were banned. There was like a lot of lockdowns. So the virus was only progressing through like geographic land. So it's kind of like a model for with a lot of data for that period. So what you can actually observe, and you can see here from that of Waltree, most of the biggest outbreaks were kind of like really based on these three. Well, you can relate to this like just this clade. So at first glance, you can like easily see that most. So you see here, this is one of these, and then you have the other two. So you have like most of the time in this case study where we had like 7 states, you can see that in New York City, for example, most of the outbreak really clusters within that specific clade here, and even the other two are like really close. So it seems like the seeds, even though there were like a lot of infections, truth is the outbreak kind of like depended on very few clades at the beginning. It didn't really have nothing to do with this. It's like this is a more other European cluster, and this is the cluster from China. And then that also means the importance of this was because in our analysis, that means that this is when we're talking in this case study, we kind of like we can focus easily on our data set because it's the same outbreak. We can see that we like if you look at the trees from the different states, you're not referring to like in one case an outbreak for like let's say a China clade and then like another one from a German clade. Most of them keep being of that specific clade, so it's pretty much the same outbreak. Again, I'm talking about the first 6 months. And then based on this data set, you can sample from these sequences and you can build a more, let's say, a tree that connects all these different states. And then from there, you can derive the probability of having a sequence clustering with itself or clustering with a neighboring state. And you can pretty much develop a network where, in this case, you can either calculate the ingrowing rate or the outgoing or the incoming, and then you can also calculate what is the transition rate from one state to the other. So that was kind of like a network for this case that we built. Then also we were able to identify other factors that really associated with the early spread, and some of them has to do with urbanization, again, the distance from New York City, then infection rate in neighboring states, the infection rate in the same state, etc. So these are like factors, different factors that have a really high fit in the spread on the model, and then we were able to use that in our models. So we built 4 different models of different complexity. The first model was just a baseline model where we use urbanism, the distance from the hotspot, and the reproduction rate. And you can see at the beginning that you can see the different weights. And as you progress, we started to use what is called genetic connectivity in this, which pretty much are these rates that I was describing before, like how connected these states are in the network. And then based on this, we can have like each transition from one model to the other by including different measures was significant. And you can see here we're including in our full model the reproduction rate, how urban the place is, the incoming rates, the outgoing rates, the ingrowing rates, and then also the transition distance from hotspots. Based on this model, we're able to like go to that network and say what happens if we cut specific connections in that model. And then obviously you can have a much bigger effect instead of like going and like freezing everything, just by connecting, for example, here, the connection between New York and Pennsylvania, you can actually, this is the number of deaths, of the death rate you can save. Obviously, this is across different time points. So here on the second panel, you can see across different dates. So it probably means that when you're here, you can you have to intervene in these two here, like at different time points, you have different different locations in the network that make sense to intervene. And then what's interesting was that we compared these results to like a complete lockdown that we did. And what's interesting about the complete lockdown is that obviously the best way to like stop a virus is for everybody to stop moving. So like a lockdown around 0.9%, like 0.9 of success, or like 90% of success, obviously kind of like will block the network from transition. But what's interesting is if the lockdown is not as efficient, then even by including specific intervention on the map. So for example, if we only include one or two interruptions in the network, you get the same effect of like, let's say 70% success of lockdown. So in reality, what that says is that if we cut like 2 edges from that network, then that corresponds to a 70-degree success of like a complete lockdown. It's a fair question would be, how do you suggest to do that? Kind of like cutting these edges. Obviously, there's the obvious, like in some cases, to kind of like reduce the transmissibility from one place to the other. But there's also different strategies you can have. For example, vaccination. You can have like some sort of like health centers. Where do you allocate the resources for like campaign or something like that? Especially as we saw that it's tough to pretty much engage a whole country into the same common goal, but you might be way more successful if you during time of a pandemic or an outbreak. It doesn't have to be a deathly pandemic. You can actually allocate resources and be very successful in key nodes of this network. That's the approach. Yeah, actually, I feel like I was running a little. I was a little faster than we're discussing. So let me, in summary, explain what our results suggest is that all of the initial outbreak can be traced to a few lineages, indicative of a continuous initially viral flow. The genetic connectivity becomes increasingly significant later in the first wave, rather than the beginning, which is like the distance from the hotspot seems to matter a lot. Isolated local strategies like herd immunity that has been discussed often, and people don't really think about it that much, can have a negative effect on neighboring regions. So yes, you might decide that you're doing your own thing, but actually that has a direct effect to everybody that's around you. It's also works in the reverse. Everybody was praising the model that Sweden was doing, but at the same time, what they were not saying was that there was a big wall around Sweden where everybody else was doing like one of the best lockdowns ever. So Sweden, even though they had like their own ingrowing rate, there was no incoming rate from the other states as well, that they were like actually under very strict lockdowns, like Finland and Norway, etc. So Sweden, first of all, they were like pretty far away from like the viral centers in Europe, and at the same time, they did build a kind of like, not themselves, the countries around them built like a big wall that some people are fan of. Anyway, so that it matters like what you do. It matters. It impacts your the neighboring regions. So you can have a more efficient mitigation strategy if it's unified. It's kind of like with cross-border interventions, or like a few targeted internodes that can have similar results, similar to like complete lockdowns, because complete lockdown is never like and you can never have 100% complete lockdown and like for a long time, but you can actually have like surge in like specific situations more efficiently. All right, so before I get any questions, and actually I think we have 15 minutes for questions, which is great. If you have any questions, I do want to give a shout out. We're gonna get an announcement here. William also mentioned it. We're starting a biomedical data science center here at NYIT. You're gonna get the announcement from our provost very soon, and this is the affiliated faculty so far. Obviously, the center is like open to everybody. If you want to join this effort and help us grow, that would be amazing. You can reach me. I'm going to be, at least for the first year, the director of the center. And you can reach me at my emails if you have any questions about this center, or if you want to work with us, collaborate, be a part of this, like, you're more than welcome. And with that, I will take any questions if you have about the talk. I hope it wasn't like too specific, like you could get like a little bit of a bigger picture as well.
Right, thank you, Leonidas. That was really great. Does anyone have any questions? As Leonidas just asked. So. Well, I have one, Leonidas, if to start things off.
Sure.
So I was interested about back to your tumor evolution model. Couple of things. So if I understood it correctly, you're just looking at one tumor, right? And based on the variant frequencies, you're able to get a sense of the time ordering of events.
Yes.
So where then are you measuring the growth rate? Because you're looking at one tumor. So how are you saying growth? Are we looking at like the number of mutations?
So I did two things. So at the beginning, I applied this to like about 1,000 tumors, but I also was able to, the nice thing about this model is that it can be applied in a single tumor. So I can, it becomes a little more complicated, but what happens is that, whenever you have all these mutations that happen, let's say before the driver, I can use a window of these mutations, like the var-allele frequency that they have and how much
a window around the frequency.
Well, let's say if the third one, like in this case, it's a simplified version where, sorry, let me see. This is a simplified version where you have, let's say you have this mutation that happens here as a driver, but in my model, I will use, let's say 150 of mutations that happen before this one, or 100. It depends on how well the tumor is sequenced. That kind of like the blue and the red one, which we consider hitchhikers. So at the end of the day, I'm taking a sample of the hitchhikers and think of this as a sliding window. And then every time I can estimate what is the tumor doing? Is it growing? Is it like how fast is growing? And then based on that window, I can estimate the impact of the mutation that happens next after that window in a way.
If a lot of mutations have the same frequency or this very close frequencies, you say that's a high hitchhiking, high growth rate?
Yeah, they seem to, like, if you see here, actually what happens is, let's say this is kind of like the frequency of the driver mutation, but it's pushing up also the hitchhiker mutations. So think of this as if I have 100 of this and see, kind of like study how it's pushing up the hitchhiker mutation from what you were expecting in a way. So this would be expected if there's no driver, and then you can see that now you have all of this kind of like growing in time. So pretty much when I'm gonna get when I'm gonna get the sequence mutations, then I can see what is the difference between this and this. What is the difference between this and that? So kind of like in a generational step, how close is the frequency of these generational mutations? So here you can so you have a delta one from, let's say you have the final blue frequency and the red one, but then you'll have a modified delta for a generational mutation if it's actually is being pushed up by a driver. So this is going to be reduced than what you're expecting. So based on this kind of like reduction, I model this kind of like change and be able to see how much they're being pushed and how much how fast the tumor is growing. Actually I don't have. it becomes a little, it looks a little more complicated that there is, but this yellow part represents the cell growth without the fitness mutation. And then you have an additional part, which is the extra cell growth with the fitness mutation. And then you can pretty much, for each mutation, you can calculate the growth and the effect.
Oh, I see. So there's some
people.
parameters in here that we have to estimate first.
Yeah, I didn't want to go to the
Okay, yeah.
you know, it's
No, but I still think it's really neat. It's
what is cool about this is like I have also included like what is called a generational time. So every time you take a window, you're sliding the window, it gets reset. So whatever happens later does not really depend on what happened earlier. So it's not gonna, like, if something happened at the beginning of the tumor, it's not gonna scramble the signal later. So every time I calculate this window, it gets reset in generational time. So I don't have to be dependent so much on previous estimations. Yeah, so that was the, yes. I don't know, does that make sense? So
I think so, yeah.
You have here, you order the VAFs and then you calculate the difference. And then obviously it's going to be very unstable to go from one mutation to the other. But if you take a chunk of them, then you kind of like decrease the noise that you have. If you get 100 of them, there's like a different expectation based on the generation time.
Yeah, that's an interesting way of looking at it. I was wondering, do you also see any relationship of the driver mutations to predisposition variants, like and to risk SNPs that people have before they get cancer?
I mean, you can study that to see.
Yeah,
it seems like you can look at the really early. This is kind of like, I think you're referring more to this part, but it's like look at the early versus late mutations.
Or like even before cancer starts, there's predisposition variants, right? So
yeah, but if then that these mutations would appear as like and reads like as an early mutation, because even if cancer starts later, these mutations will continue to exist in the cell, right?
Yeah.
You're never, like the ones that happen earlier, you're never going to wash them in the same way. If you look at, let's say, a population that is expanding here, the blue one that happened at the beginning is going to exist in the majority of the population in the higher frequency as well at the end, right? Because even though this is not a cancerous mutation, because it happened early, it will always have a higher frequency than the fitness mutation that happened later because it pre-existed, right? And that's kind of like what I'm using to model. So in that way, you could say that the ones that seem to, like, you know, are pre-cancerous in the way you want to define it would be more highly enriched as an earlier mutation.
Okay.
You can certainly, if you have like a group, you can say that group of mutations are enriched. So here, for example, I have specific ones, for example, the cancer pathway, cell cycle, apoptotic mutations, immune response mutations were also positive at the beginning. So, you know, you can look at different groups and see if they're like early versus late in that sense.
Yeah, interesting, neat.
Yeah. Any other questions?
Spencer's got one.
Oh, hi, Spencer. Yes.
Hi. I just have a quick question in terms of the sampling of the tumors over time. Are we talking about taking multiple sequential samples from the primary tumor site or from secondary tumor sites, or just randomly, or?
No, so there's no, so that was what I thought was cool about this, is like you're not really sampling across time because you're not going to sample the tumor across time. What you're doing is pretty much get a screenshot at the point that you're sequencing. So you're not going to, you're trying to pretty much project and extrapolate on the tumor growth and the impact of mutations, even though what you have is a screenshot at the sequencing time. So you get all of these VAF, the frequency of all of these mutations, and based on the frequency of all of these, you try to pretty much reconstruct how the tumor was growing, has been growing, etc. So there's no, when we have here A, B, C, this is when the mutation happened, but you're only sampling during sequencing. You're not, and that's because you like, you know, when somebody has cancer, you cannot pretty much say, all right, let's please come back in a few months, and then in another few months and see how that's going. So you only have one chance to get the mutation frequencies. You could do with PDX mice, but there are some questions there that has to do that they are like immunocompromised. So the validity of these results might, you know, like be debatable to some level when you're doing with some specific tumors. Does it make sense? Sorry, did I answer the question?
Yes, you answered the question.
Okay.
Anyone else? Any students want to venture a question? We have a few student guests today.
All right, so yeah, if you're interested in this, please reach out, or if you're interested in the new center, again, please reach out. So we're pretty optimistic, and you know, then you can help us grow it.
Yeah,
I think it hasn't officially started yet, but it should, like, you know, in the next few hours, I'm expecting, or days.
Okay.
Yeah.
Well, anyway, well, that's great. Well, thank you again, Leonidas, and thank you everyone for joining us today. This was our last talk for the semester, but we look forward to joining you again in the spring with another round of exciting talks from New York Tech faculty. So don't forget to check out our Canvas course and for archives on any of the previous talks. And that's it. So take care, everyone. Have good luck with your final exams, students and faculty. And have a good rest of your day and a fantastic winter break. We'll see you in 2024.
All right, thank you, Will.
Thank you.
Thank you, everybody, for coming. And have a great, like, you know, happy holidays.
Thank you very much.
Thank you.
Thank you.